Welcome to today's session. Myself Jalaja Sajeev, PGT Bio from KV Keshapuram. Our today's session is regarding multiple allelism. First, see the heading multiple allelism. Multiple means many. So, it is the occurrence of many alleles that controls a particular character in an individual of a population. Each allele produces a different phenotype. Multiple alleles are occurring in the same locus of the homologous chromosomes, and all these alleles, they are the mutant forms of the same gene. Moreover, they affect the same character, same trait. So, multiple allelism is the occurrence of many alleles affecting a particular character seen in different individuals of a population, and they are produced by the mutation of a wild type allele or a dominant allele. They produce different phenotype. They are seen in the same locus of a homologous chromosome, and they will affect the same trait or character. Example, ABO blood group system in humans. You know, ABO blood group system, it was discovered by Landsteiner. AB blood group was discovered by De Castello and Steeny in 1902. Blood grouping, we can group the blood into A group, B group, AB group and O group. So, this blood grouping, that is based on the presence of antigens present on the surface of RBC. And you know, antibody that is present in the serum, you know, serum is the fluid part of clotted blood, and plasma, that is the fluid part of unclotted blood. As I have already told you, here there are the presence of antigens, and these antigens, they are present on the surface of the RBC, which are responsible for producing different blood groups, such as group A, then blood group B, AB blood group and O group. Now, for producing a particular antigen, there needs alleles, and the different alleles are IA, IB, IO. This IO allele, it can be represented by using recessive I, small letter I. Small I can also be used because it is the recessive allele. Now, I stands for, why we are using the letter I? I stands for immunoglobulin. Immunoglobulin begins with I. So, this particular letter we are representing here for the alleles. So, I stands for immunoglobulins. IA allele, when we are observing, it is dominant over IO allele, and similarly IB allele is dominant over IO allele. Now, comes to these two alleles, IA and IB alleles, they are showing codominant. They are codominant and they show codominance. Already we have discussed about this codominance. So, these two alleles IA and IB both will contribute equally in the production of the new phenotype. And now coming to IO allele, or it can be represented by small I. And this particular IO allele is the recessive among IA allele and IB allele. So, IA is dominant over IO. IB is also dominant over IO allele. And IA and IB alleles are codominant and IO allele is recessive. Now, when we consider this ABO blood group system, here there is the presence of 4 different types of phenotypes and 6 different types of genotypes. Very important point, 4 different phenotypes and 6 different genotypes. Here phenotype means blood groups, that means A group, B group, AB group and O group. Blood group A is produced by the alleles I, A allele. So, A allele that will produce A blood group in homozygous as well as heterozygous condition. B blood group that is produced by IB alleles again in homozygous condition or in heterozygous condition. Now, coming to O blood group. O blood group is produced only by IO allele. Again, it is produced only in homozygous condition because it is the recessive allele. Now, coming to AB blood group. AB blood group is produced by IA allele and IB alleles together because they will produce AB blood group and it is an example of codominance. A allele and B allele will equally contribute in the production of AB blood group. So, it is an example of codominance. Here you can see the representation, and there is another character, that is rabbit coat color, which is also an example of multiple allelism. Similarly, there is another one, that is the eye color in Drosophila. Among Drosophila, the wild one, that is red. The different eye colors are red, blood, eosin, coral, cherry, apricot, honey, buff, trenched, pearl, ivory and white. So, a series of eye colors are there in Drosophila melanogaster, that is also controlled by a sequence of multiple alleles. So, here in the ABO blood group system in human beings, you can see here the different phenotypes, that means the different blood groups, blood group A, group B, AB blood group and O blood group. Now, see here you can see the allelic constitution. A blood group can be produced in two genetic constitution. One is IA allele, A allele in homozygous condition. Here this is heterozygous one, IA allele and small I allele or IO allele. So, this will result in the production of A antigen and hence the group is A group. Here B blood group produced by two genetic constitution, one is in homozygous condition of IB allele, that means IB and the other is heterozygous condition, that means IB and IO. So, these combinations of alleles, they produces B antigen. Now, coming to AB blood group, there are both the alleles, IA allele and IB alleles. So, there is the production of A antigen and B antigen. So, there is the group AB blood group. Now, coming to O blood group, it is produced by IO alleles in homozygous condition. So, represented as IO and or we can represent it as small I because it is the recessive among all the alleles. Now, coming to the antigens here, A blood group has A antigen, B blood group has B antigen, AB blood group has A and B antigens and O blood group without any antigens. Here again it is the same thing, you can see here. Now, at another example for multiple allelism is coat color in rabbit. Black, it is controlled by, it can be represented as capital C gene. Capital C will produce black coat color. CH, alleles, allelic representation in homozygous condition produces the coat color chinchilla. Now, the other coat color, that is Himalayan, it can be represented by the allelic constitution CH like this. And you see this albino, pure white, this is controlled by recessive gene. So, wild one, dominant one, the most dominant one that we will say it as wild. Capital C is dominant over CH allele, which is dominant over CH allele. Again it is dominant over small c allele for albino. Now, comes to the at another aspect, chromosome theory of heredity. It was put forward by Sutton and Boveri. According to them, pairing and segregation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united this chromosomal segregation with Mendelian principles. This theory, it states that chromosomes are the vehicles of inheritance as they carry the hereditary factors or genes, and these hereditary factors or genes, they are transferred from parents to offsprings through successive generations through chromosomes. So, in short, chromosomes are the vehicles that carries the hereditary factors or genes which are transmitted from parents to offsprings through successive generations. Chromosome theory of heredity that was confirmed experimentally and proved by Morgan, Stuyvesant and Bridges. They established the fixed and definite location of genes, position of genes in the chromosomes. This particular theory, the chromosome theory of heredity, it forms the basis of modern genetics. Now, coming to yet another aspects linked to this chromosome theory of heredity, that is the parallelism between genes and chromosomes. Both genes and chromosomes are seen as pairs. You know, each and every character in an organism is controlled by a pair of factors, genes. And similarly, chromosomes are also seen as homologous pairs during the zygote stage of meiosis. And each member of this particular pair, that is contributed by the two different parents. So, in a pair of homologous chromosomes, one chromosome is contributed by father and we will call it as paternal chromosome, and the other is contributed by mother, so known as maternal chromosome. Genes remain unaltered in the subsequent generations. Both will segregate at the time of gamete formation. So, the individuality of the chromosomes as well as the genes, they are remaining intact, it will not segregate. And during gamete formation, these genes, gene pair as well as homologous pair of chromosomes, they will segregate and move to two different gametes. Segregation of one pair of genes or alleles or chromosomes, that is entirely independent from the segregation of the second pair. Now, after gamete formation, by the union of gametes during fertilization, these paired condition of homologous chromosomes as well as the paired condition of factors, genes, alleles are also regained. So, it is regained by the process of fertilization. Now, in most of these cytological and genetical studies, there is the use of Drosophila melanogaster. Why it is used widely as an experimental material? There are so many reasons. One, it can be easily grown in the laboratory. Second, it produces more number of progenies. Third, it can be grown in minimal nutrient medium in the labs. Fourth one, male and female can be distinguished very easily. The next point, limited number of chromosomes. That means 2n is equal to 8 for pairs of chromosomes they have, and each one has separate distinguished characters. It can be easily identified with the help of microscope. Can easily identify these 4 pairs of chromosomes with the help of microscope. So, and it is easily available. This material, that is Drosophila melanogaster, is easily available. It is known as fruit flies. Here are some of the different types. Wild type, the dominant one, it shows dominant characters. Here with the red eye color and large sized normal wings. This is ebony body, again red eye color, normal wing size. Here you can see the size of the wing is very less, miniature type, or we can call it as vestigial wings, and body color yellow body color. Here you can see curled wings, eye color is red, curled wings. So, these are the different types of Drosophila melanogaster. Now, come to the topic here, linkage and crossing over. Linkage, linked together, seen together, attached together, and remain together. So, it is the tendency of the genes to present together in the same chromosome, and these genes, linked genes, they will always remain together and they will travel together from one generation to the other, from parents to offsprings. They will never segregate. That association of linked genes, it will remain together. So, this particular phenomenon, that is known as linkage, tendency of two genes to remain together and travel together from one generation to the other. And the genes that are seen together and inherited together from the same chromosome, it is known as linked genes. Here are some of the examples for linked genes. The gene controlling red eye color and grey body color in the case of Drosophila, and these two genes controlling two different characters, but it will remain together in the same chromosome, and it is transmitted together from parents to offspring. So, such genes, they are always remain together in the same chromosome and travel together from parents to offspring, is known as linked genes, and this particular tendency, that is known as linkage. Here you can see these two genes, gene 1 and gene 2. These two genes, they are seen very closely located. So, there is no chance of crossing over in between them. They will not separate. So, such genes, they are seen very close to each other in a particular chromosome and remain together in the same chromosome and travel together from parents to offspring. They are known as linked genes. Here you can see they are seen in different chromosomes. They are not linked. So, here there occurs the chance of crossing over between these genes. So, these two also you can see gene 1 and gene 2, they are seen at a greater distance. So, here in this case there is the chance of crossing over between these genes and hence they will get separated. So, these are not linked genes, but here in this first case it is linked gene. Unlinked genes, they are located on different chromosomes and they assort differently and give a dihybrid ratio. You know the dihybrid ratio 9:3:3:1, but this is given by unlinked genes and it shows dihybrid normal test cross ratio. What is the dihybrid normal test cross ratio? Yes, 1:1:1:1. But come to these linked genes, you know they are located in the same chromosome and they do not assort independently. There is no chance of crossing over between them. And most of the time they will be seen very close to each other. So, there is no chance of crossing over. And as a result, even though they are two different genes contributing to two different characters, they will produce a dihybrid ratio 3:1. This is an amazing fact here. Normal ratio, dihybrid ratio for unlinked gene is 9:3:3:1. Now, for linked genes, that is the difference. Here comes the difference. Linked genes will produce a phenotypic ratio 3:1. You know they are controlling two different characters. So, it is an example of a dihybrid cross, but dihybrid ratio it will be 3:1 instead of 9:3:3:1. And similarly, these linked genes they will give a test cross ratio 1:1. You know normal dihybrid test cross ratio is 1:1:1:1, but it will behave just like that of monohybrid test cross. You know the monohybrid test cross ratio 1:1. So, the same ratio 1:1 will be the test cross ratio for linked genes. It is quite interesting and amazing. Now, come to some examples of complete linkage. In Drosophila, melanogaster, homozygous red eye is dominant, wild one, and normal winged. So, both these characters are dominant ones. So, it is represented as wild type, dominant, the most dominant one. Such a female fly that is crossed with a double recessive male. In the place of red it will be white eye, and in the place of normal wing the recessive character means vestigial wing or miniature wing. The F1 flies, in such case all the F1 flies are heterozygous and they have the normal dominant phenotype. That means red eye and normal large sized wings. This F1 dihybrid fly is then test crossed with the recessive male. You know test cross, F1 hybrid crossed back to its recessive parent. Here the male individual is recessive. The F1 dihybrid with red eye and normal large sized wing is crossed with its recessive parent with white eye and miniature wing. The progenies comprises only two types of flies. Red-eyed normal wings 50% and white-eyed and vestigial wings 50%. That means test gross ratio becomes 1:1. No recombinants are produced here. If there is chance of crossing over, then there will be the production of 4 different types of gametes. Then 4 different types of phenotypes will be obtained. But in this case, since they are linked genes, they are not segregating. So, they will remain together in the same chromosome and transferred to the next generation. So, here there is the production of only 2 types of phenotypes, and hence in this test cross we got 1:1 ratio only. So, no recombinants are produced. That means only parental types are produced. Red-eyed and normal winged one 50%, white-eyed and vestigial winged one 50%. Both are the parental types. So, the reason is there is no crossing over between linked genes. It is a fact. No crossing over, and hence there is no production of crossovers or recombinants in linked genes. Morgan later crossed a white-eyed miniature winged female Drosophila with red-eyed and normal winged male. In the F1, one red-eyed and normal winged female is produced and one white-eyed miniature male is produced. When these F1 females are intercrossed to produce the F2 generation, 3 red-eyed and normal winged, that means dominant progenies, 75% normal dominant progenies are produced in the F2 generation here, and 25% that will be white-eyed and miniature winged progenies are produced in the F2 generation. That means even though it is a dihybrid cross, here the phenotypic ratio varies. It varies from 9:3:3:1, the normal ratio, to 3:1. So, this shows the variation of linked genes and unlinked or non-linked genes. Now, coming to linkage group, all the genes attached to, linked to a particular chromosome, that is known as, that group of genes, they are known as linkage group. Since the genes in a particular chromosome have its alleles in its homologous chromosome, we will consider this pair of homologous chromosome as a single linkage group. Very important concept. In the homologous chromosome, linked genes have its alleles. So, we can consider this homologous pair of chromosome as one particular linkage group only. So, when we consider like this, in the case of human beings, you know, we have 23 pairs of homologous chromosomes. So, these 23 pairs of homologous chromosomes, we can call it as 23 linkage group. So, what is the linkage group of human beings? It is 23. If it is 23 in human beings, you know, in Drosophila, how many chromosomes will be there? 2n is equal to 4 pairs. So, the linkage group of Drosophila is 4. Like that, maize plant has 10 linkage group. 10 linkage group means it possesses 10 pairs of homologous chromosomes. Garden pea has 7 linkage group, means it has 7 pairs of homologous chromosomes. Traits in Morgan's crows. Morgan conducted some experiments in Drosophila by considering some of these characters. He considered a yellow body color and the other one, that is grey body color. Grey body is dominant over yellow body. And here you can see white eye color is recessive and the dominant eye color is red eye color. Now, similarly wing, miniature wing or small sized one, that is known as vestigial wings. Recessive character, recessive allele, and the other one, that is normal sized normal wings, that is represented by this M plus. The wild alleles are dominant and they are represented by plus. Here it is grey body represented as Y plus. Red eye represented as W plus. Normal wing represented as M plus. Now coming to Morgan's experiment with Drosophila. Consider the character white eye color gene, recessive one, represented by small w here, and the miniature wing represented by the first letter small m, again recessive character, we can call it as vestigial wing also. And both of these genes, they are located on the X chromosome. They are located together in the X chromosome. So, Morgan in 1911 crossed a female white eyed and having miniature wing or vestigial wing fly Drosophila with a wild type of Drosophila male with character red eye and normal sized wing. In the F1 among the progenies, all the males are white eyed with miniature wing, white eye and miniature wing, and here it is Y chromosome. It does not have any allele for these characters, and all the females were wild type. Wild type means again red eye color and normal wing size. Here you can see capital W or small w and plus, and here it is capital M, it shows large sized normal wing. So, in the F1 all the females are normal and wild type with red eye color and large sized normal sized wings. Now, the possibility of the gametes. F1 progeny that produces these different types of gametes with alleles capital W and capital M together, small w and small m together, but here these are the two conditions, one and only when there is crossing over. That is small w and capital M by crossing over, and here it is capital W and small m by crossing over. That means if the distance between these two linked genes is more, then only there is the chance of crossing over and production of crossovers or recombinant gametes, and thereby the production of recombinant progenies. So, 25% each if independent segregation is true, but again the chance is that the occurrence of crossing over between linked genes is negligible. It depends upon the distance between the two linked genes in the chromosome. If the distance is more, then there is chance for crossing over, but if the distance is very less, then there is no chance of crossing over. Now, Morgan got in the F2 generation the different types of progenies: white miniature, white normal, red miniature, red normal. That means here also again it is only in the case of crossing over. The progenies can appear in 1:1, 1:1 ratio. Otherwise, I have already told you, if there is no crossing over between the linked genes, progenies will be appearing in the ratio 1:1 in test cross and 3:1 in F2 generation. In F2, the most frequent phenotypes for both the sexes were phenotypes of the parents of the original cross. That is white eye and miniature wing, one parent, recessive parent, and the other red eyes and normal wings, wild type. Non-parental phenotypes. Non-parental phenotypes means recombinants. That means white eye with a normal wings. Similarly, red eye with miniature wings. They are produced only by crossing over. Well below the 50% predicted for independent assortment. If there is independent assortment, then there will be parentals and recombinants, all are appearing in equal proportion, 1:1, 1:1. But here it is well below this 50%. That means parentals are appearing in larger quantities than the recombinants. Recombinants, the chance is very less, negligible numbers. During meiosis, alleles of some genes assort together because they are near each other on the same chromosome. Recombination occurs when the genes are exchanged between X chromosomes of the F1 females. Since these linked genes, here in this particular case it is present on the X chromosomes of this individual. So, between these genes, if there occurs crossing over, then only the chance of production of recombinant is there. Now, linkage in sweet pea at another character that was studied by Batson and Punnett in the year 1905. Traits, it is present in sweet pea. The different traits are flower color, it may be either purple or it may be red. Purple is dominant over red color. And the second character studied was pollen shape. It may be oblong or it may be round, but oblong is dominant to round character. Here Batson crossed a purple and long one. A sweet pea plant producing purple flower and long pollens or oblong pollens crossed with another one producing red flower and round pollens. A red flower and round pollen, recessive character. Among the progenies F1, it was seen that all the plants are having purple flowers and long pollens. In F2 generation, that is by the selfing of F1 hybrid progenies. Here you can see the production of 4 different types of gametes. Capital P and capital L for purple and long, capital P and small l purple and round, small p and capital L for red and long pollens, small p and small l for red and round pollens. F2 possesses 4 different phenotypes. In the F2 generation, the observed ratio, he got the ratio 15.6 is to 1 is to 1.4 is to 4.5. What is this? Here the ratio is varying from this expected 9 is to 3 is to 3 is to 1. This is an example of a dihybrid cross. We know in dihybrid cross the ratio must be F2 ratio, F2 phenotypic ratio must be 9 is to 3 is to 3 is to 1. But here it varies. What he observed here is 15.6 is to 1 is to 1.4 is to 4.5, entirely different. So, this 9 is to 3 is to 3 is to 1 normal ratio of F2 generation, normal dihybrid ratio, this was expected, but it is not seen. Here in this case, Batson observed parental phenotypes. Parental phenotypes were more than non-parental phenotypes. You see here 15.6 parental type and 4.5 again parental type. Now see the case 1 is to 1.4. There occurs a slight change in this case, a chance of crossing over. So, limited number of, very less number of crossovers are produced here. Parentals we can see in large quantities compared to recombinants or crossovers. So, it shows that these two genes, they do not assort independently as in the case of dihybrid cross. And these genes are physically linked on the same chromosome. And the distance between these genes will be negligible. Now, some key points regarding linkage. Linkage between genes is detected as a deviation from the expectations based on Mendel's principle of independent assortment. The frequency of recombination measures the intensity of linkage. In the absence of linkage, this frequency is 50%. For very tight linkage, there is no crossing over, it close to 0. Recombination is caused by physical exchange between paired homologous chromosomes, early in the prophase stage of the first meiotic division, after these chromosomes have duplicated. So, crossing over takes place in the Pachytene stage of meiosis. During zygote stage, the homologous chromosomes will pair each other and and that phenomenon we will call it as synapsis. They will form synaptonemal complex. At any one point along a chromosome, the process of exchange, that means crossing over, it involves only 2 of the 4 chromatids in a meiotic tetrad. Late in prophase 1, crossovers become visible as chiasmata. So, the region of crossing over, that is known as chiasma singular, chiasmata plural, and crossing over we can define it as the exchange of segments of non-sister chromatids of a homologous chromosome. It takes place in the Pachytene stage of meiosis, and the point of crossing over is known as chiasma or chiasmata. Number of crossing over equals number of chiasma formation, and after crossing over we will get the parentals as well as crossovers or recombinants. Now, coming to the significance of crossing over, it helps in the production of new combination of characters or traits, which results in the production of variation and production of new species. Since there is variation, it leads to evolution. It helps in chromosome mapping. The position of genes in the chromosomes can be mapped based on the percentage of crossing over, and it helps in establishing the linear arrangement of genes in a chromosome. Now, here you can see some differences between this monogenic inheritance and polygenic inheritance. Monogenic inheritance, it is regarding qualitative traits or characters. In polygenic inheritance, we have studied about quantitative traits. Monogenic inheritance, where the characters are controlled by single pair of contrasting genes. Here it is controlled by many pairs of non-allelic genes. Phenotype shows discontinuous variation. Here phenotype shows continuous variation. No intermediate phenotypes are produced in this case. Here in quantitative inheritance or polygenic inheritance, you can see the production of intermediate phenotypes. Now, here I will give a little bit information about this thalassemia. You can see thalassemia is just like that of sickle cell anemia. It is controlled by autosomal recessive gene. So, it is also an autosomal recessive disorder. The cause of this one is either due to mutation or due to deletion. What is the effect of this mutation or deletion? It leads to reduced rate of synthesis of the globin protein needed for the formation of normal hemoglobin. So, since there is a reduced rate of synthesis of this globin chain, there will be the production of abnormal hemoglobin. And coming to the types of thalassemia, there are two types, alpha and beta thalassemia. Alpha thalassemia caused either due to deletion or due to mutation of HbA1 and HbA2 genes present on the 16th chromosome. And beta thalassemia, it is caused due to the mutation of HbB gene present in the chromosome 11. Now comes to the difference between thalassemia and sickle cell anemia. You know, thalassemia is a quantitative problem, whereas sickle cell anemia is a qualitative problem. In thalassemia, there is reduced rate of synthesis of globin protein needed for normal hemoglobin formation. Reduced rate means the amount of hemoglobin is less here. Here in sickle cell anemia, it is a qualitative problem where incorrect hemoglobin is produced or abnormal hemoglobin is produced, which leads to change in the shape of the RBC, the quality of the RBC. It is a qualitative character. It becomes sickle cell. Thank you.